Hello friends! Welcome back to the channel, or if you're new here, a warm welcome to you too. I hope you're all doing great. Let's jump into today's lecture. There are 7 types of asexual reproduction starting from first, that is binary fission. Second is budding, which covers budding in yeast, budding in animals, exogenous or external budding, endogenous or internal budding and strobilation. Third, fragmentation. Fourth, gemmation. Fifth, regeneration. Sixth, spore formation or sporulation, which have further types. That are zoospores, conidia, chlamydospores, oidia, and sporangiospores. 7th types is vegetative propagation, but hold up, we'll talk about this in a separate video because it's a huge topic. Alright, so let's break it down one by one. Let's start with budding. No, we're not talking about flower buds, although those are pretty cool too. In biology, budding is a form of asexual reproduction where a new individual forms as a small outgrowth or bud on the parent organism. A bud is a small outgrowth on the parent organism that develops into a new individual through the process of budding. The bud then grows, develops, and eventually detaches to become an independent organism. Think of it like a biological copy-paste of the parent. It occurs in yeast and animals. Let's discuss that. Budding in yeast. In yeasts, which are unicellular fungi, budding is the main form of reproduction. A small bud forms on the parent yeast cell, and as it grows, it separates, becoming a new cell. Imagine a tiny mini-me growing on the parent. These new yeast cells can keep growing and dividing in favorable conditions, basically making the yeast population grow like crazy when there's a lot of sugar around.
Remembrer, fermentation and baking.
Sometimes yeast may bear many buds which may further bear daughter buds. This budding stage in yeast resembles a genus Tarola. Therefore, this condition is called the Tarula stage, and the process is known as torulation. Now, let's look at budding in animals. Animals can also reproduce through budding, but this is more common in simple animals. Let me break it down for you. First, exogenous or external budding. In external budding, the bud forms on the outside of the parent. This is what happens in creatures like Hydra. Little buds appear on the body of the Hydra, and after some time, they mature, eventually breaking off to become independent creatures. It's like the Hydra is growing little mini-me's on the side. Sometimes it remain attached and become a more or less independent member of the colony, as in Psychon. Exogenous budding also occurs in certain annelids such as Psyllus and Urochordates or tunicates such as Salpa. Now let's look at endogenous or internal budding. Now, in internal budding, the bud forms inside the parent organism. Some animals like freshwater sponges like Spongilla and few marine sponges reproduce in this way. The internal bud is formed inside the parent body, which is called as gemmule, meaning internal bud, and it can survive harsh conditions. Gemmules consist of small group of cells called archaeocytes enclosed by a protective coat. These buds grow inside, and eventually when favorable conditions come it gets break free to start a new life through micropyle, and later on forms a new colony. It's like the parent is creating backup plans, just in case life gets tough. Now, let's look at third type of budding, which is strobilation. This is a pretty wild form of budding, found in some marine animals like jellyfish. In strobilation, the parent produces a series of buds, one after the other, in a stacked formation. Think of it like a conveyor belt of babies. Eventually, each segment breaks off and becomes a new jellyfish. And they don't stop. It's like a jellyfish factory. Strobilation can be defined as repeated formation of similar segments by a process of budding. The segmented body is called strobilia or Cyphistoma larva. Each segment in the body is called ephra larva, as found in Aurelia, which is a coelenterate. The ephrae breaks at intervals. So one by one the distal ephrae are pinched off from the parent strobilia and swim in the water. The free ephrae feed, grow, and in due course of time change into jellyfishes. About a dozen ephrae are formed in a single strobilation. Strobilation also occurs in neck of Taenia, or also called as tapeworm.